A healthy human cell follows a highly regulated life cycle of dividing, repairing, and eventually undergoing programmed senescence. But in Hutchinson-Gilford Progeria Syndrome, or HGPS, that biological clock accelerates to a devastating speed. Children born with HGPS appear healthy at birth, but by age 1 or 2, they show signs of rapid aging. They experience hair loss, lose protective body fat, and develop severe joint stiffness. This process is so aggressive that these children suffer from cardiovascular disease usually seen in the elderly, leading to an average life expectancy of just 14.5 years. This condition is incredibly rare, affecting roughly 1 in every 20 million people, and unlike many genetic diseases, it is not passed down from parent to child. It arises from a spontaneous mutation, a random biological glitch that occurs at the moment of conception. Because HGPS compresses the biological changes of decades into just a few years, it provides a window into the core structural failures of human aging that are typically hidden by the slow pace of a normal lifespan. The entire disease originates within a single gene known as LMNA. In a healthy body, this gene provides the instructions for LMNA, a protein that builds the rigid scaffolding holding your cell nuclei together. To see the source of HGPS, you have to look at the human genome in its entirety. Out of the 3 billion DNA letters that make up your genetic code, a single letter C is accidentally swapped for a T. That one typo creates a cryptic splice site. It acts as a false signal, tricking the cellular machinery into misreading the genetic instructions and cutting the protein blueprint in the wrong place. That microscopic typo initiates an irreversible systemic collapse of the entire body's physical architecture. Because the cellular machinery misinterprets the genetic instructions, it fails to produce healthy LMNA. Instead, it manufactures a broken, highly toxic version of the protein called progerin. Progerin has a fatal flaw. It retains a fatty lipid tail known as a farnesyl group. In healthy proteins, this tail is temporarily used to guide the protein to the cell membrane and is then snipped off. In progerin, the tail cannot be removed. It acts as a permanent molecular anchor. Because it is permanently anchored, progerin exerts constant tension on the edge of the nucleus. It physically pulls the nucleus out of its normal spherical shape, warping and buckling the cell wall in a process called nuclear blebbing. Once the nucleus is deformed, the cell's internal architecture is compromised. The systems responsible for repairing DNA break down entirely, forcing the cell into premature senescence, where it stops functioning and dies. To treat HGPS, doctors cannot just manage the systemic symptoms. They have to physically intercept the toxic progerin protein before it can tear the cell nucleus apart. The first major countermeasure against HGPS arrived in the form of Lonafarnib, known commercially as Zokinvy. Lonafarnib is a farnesyl transferase inhibitor. It acts as a chemical shield, preventing the sticky farnesyl anchor from ever attaching to the progerin protein. While Lonafarnib extends the lifespan of children with HGPS, the toxic protein is still being produced, meaning it is not a complete cure. Researchers are now moving to next-generation interceptions. New molecules like Progerinin are being developed to bind directly to the toxic protein, attempting to neutralize its ability to interact with the cell membrane altogether. Going a step further, scientists are engineering RNA scissors. These synthetic molecules act like molecular masking tape, physically docking over the faulty splice site so the cell's machinery ignores the mutation and reads the instructions correctly. These therapies mark a shift in modern medicine, moving from the treatment of organ failure to outsmarting the cell's faulty chemical instructions in real time. But the final frontier of progeria treatment abandons protein interception entirely. Instead, scientists are turning to CRISPR-Cas9 base editing to fix the root DNA. Rather than treating the toxic byproduct alone, a base editor is designed to scan the living DNA strand, locate that specific error, and physically swap the mutant T back to a C. Backed by the Progeria Research Foundation, this technology is currently pushing towards clinical trials. The goal is a definitive, one-time cure that permanently corrects the genome of a living patient. If successful, this marks a medical threshold, transforming a fatal genetic diagnosis into a correctable typographical error. There are only about 400 children known to be living with HGPS globally, Yet the research required to save their lives has profound implications for the billions of healthy humans on Earth. Biologists have discovered that as healthy human cells age, they also begin to produce tiny amounts of progerin. This slow accumulation of the toxic protein drives the everyday atherosclerosis and heart disease experienced by the elderly. The efforts to stop progerin from damaging the cell nuclei of children are providing the tools needed to address the same protein damage in the elderly. Neutralizing this one defect in the rare few could lead to a way to prevent heart disease in everyone else. To learn more about the leading edge of genetic medicine, leave a like, subscribe, and ring the bell to stay updated with computational genome biology.